Hi everyone, welcome to my channel Pure Biology, and today I come with another video related to the plasma membrane. That is the plasma membrane, you all know, that is the topic related to the cell biology. Okay, so I have already explained about the structure of the plasma membrane in my previous video, and so it is a very large topic, so that's why I uploaded only the structure of the plasma membrane, but related topic with the plasma membrane I try to upload in each or in a single video. Okay, so you continue obviously with my channel, and if you just this channel is helpful to you, obviously you try to subscribe and share my channel and support me your to great achievement, you can say, because without your support I cannot continue this channel. Okay, so you just try to support me. Okay, this is the topic, is the motion of the lipid molecules. Okay, motion of the lipid molecules. What is that motion of the lipid molecules? Lipid bilayer, that is not the rigid and the static structure. Okay, in case of cell wall, you all know that in plant cell, the cell wall is the rigid structure because lignin and cellulose both are present in the cell wall. But here the plasma membrane is not rigid structure, it is very static, not static, it is very elastic, you can say. The elasticity is there in the plasma membrane. So in a lipid bilayer, that lipid molecules can rotate freely. Why this elasticity is seen in the plasma membrane? That is the reason of lipid molecules movement. Clear? Lipid molecules, these are not the stable molecules. Okay, they can either rotate or diffuse laterally. Clear? Two types of movement of the lipid molecules generally seen. One is the rotational movement, another is the lateral motion. What is the rotational movement? When the lipid molecules rotate freely around their long axis, so this is called the rotational motion, and when this is diffused laterally, this is called the lateral motion. Okay, within each leaflet, you know, this is the one monolayer, this is another monolayer, this head and this is the tail. Okay, and same case, here is head, this is the tail. So this is one monolayer, this is another monolayer. This movement of the lipid molecules, that is contained within their monolayer, not towards easily. Okay, and this ability of the lipid to diffuse laterally in a bilayer indicates that it can act as a fluid. Okay, so if it can move within this monolayer, this is one kind of movement, or this can move to another monolayer, this is another kind of movement. If this moves in the monolayer, in each monolayer, then it is rotational movement is seen here. And if the diffusion or lateral diffusion from one monolayer to another monolayer is seen, that means the lateral motion is seen here. Clear? And the degree of the bilayer fluidity, that depends on the lipid composition and temperature. Okay, lipid composition and temperature is very important for the lipid fluidity. That means if temperature is high, that means more fluidity is seen there. Okay, but temperature is low, then fluidity also low there. Clear? And lipid composition is very important, because without proper composition, the movement of the lipid molecule is not seen. Clear? And the rapid lateral movement of the membrane lipid has been visualized by the fluorescence microscopy, and you cannot see the lipid molecules movement in general complex microscope. It is only seen in the fluorescence microscopy. Okay, and there is technique is used, that is called the photo bleaching, the fluorescence recovery after photo bleaching. Okay, The photo bleaching is one kind of technique, that is by this process we can totally see this lipid molecules movement, clear? And obviously in the fluorescence microscopy we have to use. And the transition of the lipid molecule from one leaflet to other is called the transverse diffusion. Okay, so here the lateral diffusion means, that means if lipid molecules is, there is another lipid molecules, so lateral diffusion means it goes to its place and it goes to its place and it is contained within the monolayer. Okay, and rotation means this is the long axis and the rotation of the head is the rotational movement. But transverse movement is what? The diffusion process obviously, but not here the lateral diffusion process. There is the diffusion process, this is called the transverse diffusion process. Clear? The lipid molecules from here diffuse to there and lipid molecules from here diffuse to there. This is the transverse diffusion. Clear? And for such motion to occur, the lipid head groups, which is polar and may be charged, must move into the hydrophobic interior of the bilayer. Okay, charged. Okay, so for such motion to occur, lipid head groups, which is polar, head group is polar and may be charged, must move into the, if this is charged particle, then move to the hydrophobic interior of the bilayer. Clear? And this process obviously needs energy. And the dynamic barrier also there, that prevents this rapid and spontaneous transverse diffusion. Okay, transverse diffusion is generally obstructed by the dynamic barrier. So this movement is not it is a very rare case. You just know that it is very rare case, this transverse diffusion, because there are many obstructions or many barriers are there that prevents this transverse diffusion. And this flip-flop process needs several hours to days and depends on the nature of the lipid and the membrane. Okay, this transverse diffusion also needs energy, and you can say that flip-flop process, this is called the flip-flop process, and this needs several hours to days and obviously depends on the nature of the lipid, and the rates are also dependent on the phospholipid acyl chain length and degree of unsaturation. What is unsaturation? That must be one monolayer. If one monolayer contains low lipid layer and this contains suppose high lipid layer. Okay, so this sometimes this is happening, then this transverse diffusion is seen. That means there is in the plasma membrane the two monolayer contains the same lipid composition, okay, or contains. Clear? So that's why this diffusion is seen in that situation when the unsaturated situation occur in the two monolayer. Clear? To make it balance in the two monolayer of the lipid bilayer, this transverse diffusion is seen. So this is the motion of the lipid molecules is explained here, or you can say that flip-flop process is explained here, and I will try to explain another topic in my next video. So continue with my channel. Thank you.